Thank you for coming to see my poster. I am Miharu Hara from Shibaro Institute of Technology University. This poster shows research of synthesis and evaluation of vitamin K derivatives introduced by fluorescent group NBD. I will now begin my presentation. First of all, let me explain the background and purpose of this study. Vitamin K is an essential cofactor of gamma-glutamyl carboxylase (GGCX), which is related to blood coagulation and bone formation. It's also known as a ligand for steroid and xenobiotic receptor SXR, a nuclear receptor that plays a role in drug metabolism. Natural vitamin K consists of naphthoquinone ring and side chain, and it can be divided into two types from differing of the side chain structure. Vitamin K1, phenylaquinone PK, and vitamin K2, menaquinone MKN. We recently discovered that MKN has the ability to induce the differentiation of neural stem cells (NSC) into neurons. However, the action mechanism remains unclear. To identify vitamin K binding proteins involved in the inducing neural differentiation of NSC, we have synthesized MK2 derivative 1, 2, which has nitrobenz oxadiazole (NBD) at the end of the side chain. However, the structure of the side chain is important for the induction of differentiation. So in this study, we aim to synthesize MK2 derivatives with NBD in naphthoquinone ring and compare their abilities. Next, I will explain the research method. In order to labeling of vitamin K binding proteins, we selected NBD for the fluorescent substance. The NBD has a nature of not exhibiting fluorescence in case of binding to ligand via an oxygen atom. ONBD turn off. However, when ONBD reacts with surround basic amino acids such as resin, it shifts to the protein via nitrogen and emits green fluorescence. NBD turn on. Based on this principle, NBD can fluorescently label proteins that have specific interactions with the ligand. So, the synthesis method. We chose vitamin K4 as a starting material. We introduced aldehyde at C7 of vitamin K4 in 4 steps to obtain 5. The linker of 5 was extended in the next 2 steps, including Wittig reaction to give 6. Then, in 2 steps, the C7 end of 6 was converted to carboxylic acid and the link to naphthoquinone, resulting in 7. 7 was coupling with geraniol to obtain 8. Then, 3 was obtained by introducing NBD into C7 and in 2 steps. We also synthesized 4. An amino group was introduced at the C7 end of NBDCl to afford 9. Then, 8 and 9 were coupled to form 4. The fourth is the evaluation of compounds. To confirm the efficacy of 1 to 4, we examined fluorescent labeling of SXR. MZ63 cells overexpressing SXR were cultured for 4 days and incubated with MK2 and 1 to 4 for 24 hours. After self-fixation, nuclei and SXR were stained blue by hex 33342 and red by immunostaining, respectively. Then we observed each fluorescent by fluorescence microscope. NBD's fluorescence was observed in the 4 cells for NBD probes 2 and 4. The fluorescence of all NBD probes 1 and 3 was more strongly distributed in the nucleus and outside nucleus, respectively. Once distribution of NBD fluorescent matched with SXR1, so one could be succeeded fluorescent labeling of SXR. Finally, a conclusion. To identify the proteins involved in the NSC neural differentiation by vitamin K, we synthesized vitamin K derivatives which introduce NBD in the side chain and naphthoquinone ring, respectively. We then attempted to fluorescent labeling of SXR and found that the side chain NBD derivatives fluorescent distribution of NBD was consistent with the SXR's one in the cell. This result suggests that vitamin K derivatives with NBDs in the side chain can be used to search for the proteins involved in NSC neurodifferentiation. Thank you for stopping by my poster.